All right, this is our third and our final lecture for chapter 4. We're going to look at one more way that antibodies can differ, or immunoglobulins can differ, and that has to do with what type of class they are or what isotype they have. And so this process of isotype switching, we did see in the last section that the first type of immunoglobulin made is IgM or IgD. And those are moved to the surface of a B cell, a naive B cell, and it can have IgM or IgD on its surface. But IgM and IgD, although they're the first, they're actually not the best. And so when an immune system or when an immune response is occurring, and they're good to begin with, like we need something out there right away. But once those B cell receptors are engaged, we saw affinity maturation happen, and we're going to get different types of classes of antibodies. And so we can call this isotype switching, we can call it class switching. I see them used interchangeably, so you'll see them throughout the literature in both ways. And so it is another DNA recombination event that happens, but this time the V region is left alone and the constant region is swapped out for a different constant region. Now, this is only going to occur in B cells that have been exposed to antigen. So your naive B cells are only going to have IgM and IgD on their surface, but once they get exposed, then that will initiate isotype switching. So, and the first part of this is just showing that IgM is a bound, and in its bound form, it's a monomeric. And so I was kind of talking about, I was using those phrases right away in the beginning, but IgM bound form is in a monomeric form, and it has 2 antigen binding sites. Secreted IgM is actually in a pentameric form where there's 5 different antibodies that are attached by what we call as a joining chain, or the J chain. And so there's actually going to be 10 binding sites here on a secreted IgM. Now, it's not the best antibody, but it's a really great first one to get out there. And it's strong because it does have those 10 binding sites compared to 2. So anyway, but isotype switching must occur in order for a strong antibody response. And so it is accomplished by another recombination event. But again, it's going to be in the constant region gene. So the constant segments, not the variable segments. The variable's done. The variable has gone through somatic hypermutation, and now we're going to bring in different constant regions. And we're going to do that with the switch sequences that are recognized, though, by AID. So it is the same enzyme. We don't have to have another enzyme involved. But the isotype switching then will produce antibodies that have the same antigen specificity, but different constant regions. So you can see that the variable region is the same, but rather those switch sequences will be brought together. This is looped out and removed, and now you can bring in a gamma, you can bring in an epsilon if you needed to. And so isotype switching is only going to occur during an active immune response. And what's crazy about isotype switching is that B cells can't do it alone. You might be like, why? B cell should get to decide what type of immunoglobulin it wants to make or what type of antibody it wants to make, but that's not the case. T cells are the orchestrator, and they're the ones that will tell the B cells, time to switch up that class. We need a new type of antibody out there. And we will look at that in a later chapter. Okay, but isotype switching is a big reason that, makes the antibody much better, and it's able to get places it needs to get and do things it needs to do, because IgM and IgD, they're not that great. IgD actually is pretty worthless. I'll have a meme to show you in class about that. As far as we know, who knows? Maybe IgD is like magical and we just don't quite know the extent of it yet. But it's a B cell receptor. It's It's fine. We end up changing it a lot, and then we take that D out right away. IgM, it's okay. It's big, it's bulky. It does a really good job though of activating complement. So let's just look at all those different types rather than me talk about them. We have our 5 main classes, MAG. MADGE, and I think I'll go in order there. Oh yeah, I have it listed here in order. Within those classes though, IgA has 2 subclasses and IgG has 4 subclasses, and there's probably more. This is what your text has, but again, more research is always done, and so there could be multiple classes that are listed on this slide. But overall, they all pretty much have that same structure. Looks like a Y with a variable region, a constant region, 2 heavy chains, 2 light chains. The difference between like mu and epsilon are going to have 1 more constant region. So they have a constant 4 instead of just constant 1, 2, 3, constant heavy 1, 2, 3. And largely when we talk about effector functions, so antibody is an effector protein of the immune system. It does stuff. It's a protein that goes out and does something, and so it has effector functions. And there's really 3 main effector functions that antibodies have. AB, antibody. I'll never write the full word antibody out. It's always AB. First of all, it can neutralize. It can bind up and remove toxins, virus particles, and prevent them from interacting with host cells. So neutralization is a very, very important one. Well, they're all 3 important. Opsonization. We saw how C3b was an excellent opsonin and very delicious for phagocytes in previous chapters. Antibody, specifically IgG, is also a really delicious antibody, and so it's an opsonin. And so antibodies will coat pathogen and be taken up by phagocytes through phagocytosis. Because just like we saw phagocytic cells have complement receptors, and the complement C3b was their ligand for that receptor, phagocytes also have Fc receptors, and so they can have an Fc gamma receptor, an Fc epsilon receptor, whatever it might be. Gamma is usually the one that is going to be an opsonin. And those then Fc portions, because if you think about a bacteria cell and even antibody attached, what piece sticks out and is out into the open? The Fc portion. And so the phagocytes then will recognize Fc portions of antibodies, and that will be the ligand for their receptor and cause phagocytosis to occur. And then thirdly, classical pathway of complement is activated by antibodies. And so complement activation is done mostly by IgM. And then we know the pathway that it goes, and we'll revisit that when we get to that point. But complement is activated, classical pathway, by antibodies. So we're going to break down and look at each one of the antibodies individually and talk about what they're best known for and their structure. So IgM bound form is monomeric, and then a secreted form is pentameric, where we have the J chain joining. And so it's made up of 5 different monomers bound together. So there's 10 binding sites on the secreted form of IgM. Has kind of low affinity. It's kind of an okay antibody. It's really okay to begin with and a good one to get out there, rapid response, but it's not going to be the most effective at doing the jobs. But because it has 10 binding sites, it's pretty strong binding, and complement activation is done really well with IgM. When we look at, even though it may have a low affinity, because there are 10 binding sites, that strength, we call it avidity. And so the overall binding, or the overall avidity, can be pretty strong just because of all of the binding sites. When the antigen binding portion, or the FAB, is bound to the pathogen surface, or whatever it's binding to, it will bend slightly and push out all those FAB or FC portions out into the extracellular space, right? That constant region is exposed, and then that's where complement is activated. So then we get more C3b, and you get opsonize HN there. And so a lot of pathogens can be cleared quickly by IgM. And so although it's not awesome, it's really pretty good to have as a first line. IgA is going to be created through isotype switching. And so it can only be made if T cells are like, go ahead, switch to IgA. Monomeric IgA is made, but more often it's found in a dimeric form. And so again, there's going to be a joining chain. And so the two Fc portions will join together. And so you'll end up with 4 antigen binding sites that are out exposed. The monomeric is usually going to be found in the plasma, not super high concentration, but more so the dimeric form is found in mucosal secretions. And so we see it in saliva, we see it in breast milk, we see it in sweat and tears. It's going to be lining our mucous membranes. And so these antibodies, these IgA, are just kind of hanging out, and they're really great at keeping microbiota in check. And so they're directed a lot against commensal organisms, making sure that we don't like overpopulate with too many commensals on our mucosal surfaces. IgE, this one is going to be pretty highly specialized. We see it a lot when, okay, we see it in 2 different situations. When we're looking at like an immune response, we're looking at a response to parasites or any type of, usually parasites. And so these antibodies are going to interact, and they're going to be bound actually. They're going to interact with mast cells, eosinophils, and basophils because they are going to initiate the degranulation of those cells. And so we see these in parasitic infections. The other place we see IgE, and more common in developed countries with clean and drinking water and sanitation programs, are allergies. They're bored, and so they're gonna just be like, well, let's go degranulate some eosinophils because this pollen's in the air, that kind of stuff. And so we see high IgE when we're looking at allergies, asthma, those kinds of things. IgD, I don't have a lot to say about it. It's really not that impressive. It's one of the first made, right? It's going to be bound on the surface of naive B cells. We do see it in the respiratory tract. But largely it's not a huge player in the immune system as far as we know. There are some, thoughts that it interacts with basophils, and maybe, has a role there, but largely IgD, no one really talks about it beyond it being one of the first ones made and being the, B cell receptor. Everybody talks about IgG. IgG is the king or the queen of antibodies. It is by far and away the most abundant. It's going to be found in pretty much, that's what makes up the plasma, mostly the protein part of the plasma, and the internal body fluids. It's tiny, it's pretty small in comparison to the other antibodies because it is only in monomeric form, and it only has 3 constant regions of that heavy chain, and it's pretty flexible, so it can get really anywhere. It's one of the antibodies that is able to get across the placenta, and so baby has mom's IgG circulating when it's developing. And it's just really, really good at what it does. It has strong binding affinity, and it gets better and better through affinity maturation. And so there's just a lot of things that it can do. And based on the different subclasses it has different effector functions, or most important effector functions. IgG1, subclass 1, is the most abundant and most versatile. IgG2 is going to be pretty good opsonin. IgG3 is going to be the best of IgG at activating complement. And then IgG4 is going to be really great at neutralization. But really all of these IgGs can do a lot of these different functions. So I just have them here to see that they are slightly different, but they still have the same number of segments and same number of antigen binding sites. So here's an excellent summary table of the different antibodies and their most notable functions. And you can see that IgG1 has a lot of functions. It's really great at a lot of different things, as well as IgG3. When we look at IgD, yeah, sensitization of basophils, okay. Nobody even cares about basophils though. So, I mean, people do. People do care about basophils. They're not big players in the game. So IgD is just not a huge player beyond what we see with the B cell receptor. IgM, sorry, complement activation is really what it does best because it quickly is replaced by IgG through isotype switching or class switching. IgA, you see doing a great job at neutralization. It will prevent a lot of viruses from getting past the mucosal surfaces. It'll prevent a lot of microbiota getting passed into the mucosal surfaces. And then IgE we see with mast cells. And so, yes, during an immune response for parasites, more often we see it related to allergies. Okay, I want to close out this lecture piece looking at applications of antibodies though. So outside of the context of the immune response, how have people utilized the power of antibodies? Because they are so powerful. And when we talk about antibody applications, we're really only talking about IgG. Okay, so Ig immunoglobulin, sometimes we'll call it gamma globulin because it's the gamma isotype. So you might hear gamma globulin, but it is utilized in so many different ways. So research, antisera is made by taking, traditionally, this isn't the way it's done anymore, traditionally like rabbits, horses were used to you'd immunize an animal against some sort of specific antigen, and then you'd bleed that animal and get the antisera, and then that antisera can be purified and be used in testing or could be given as a clinical pharmaceutical product. Messy, you use a lot of animals, and you end up with a lot of polyclonal antibodies that are specificities kind of all over the place. And so it's not great, but technology-wise, that was a really great starting point. Now we have a much better way to make antibodies that A, doesn't utilize animals, and B, can be monoclonal. And so you've probably heard of monoclonal antibodies, or maybe you've seen drugs that are, oops, sorry. Oh, I can turn this around, I think. There we go. And MAB, monoclonal antibody. A lot of drugs end in MAB, infliximab, infliximab, they're all monoclonal antibodies. And so this utilizes a hybridoma, which is a cancer cell fused with B cells. And so then essentially you get a cancer cell that has the longevity of, I mean, essentially could be eternal, because it's a tumor cell, it's always going to divide. And then you have a B cell that has the ability to make antibodies, have those enzymes in place to turn on those immunoglobulin genes. You can do the somatic hypermutation and all that kind of stuff. And so this is called a hybridoma. And definitely, once you get a hybridoma that's functioning, if you keep that alive, you have a gold mine, because you can just make an endless number or an endless amount of antibodies that are absolutely specific to one certain epitope. And they're all identical. They're very, very safe. And so monoclonal antibodies have really replaced like using an antisera, a polyclonal. And so clinical assays can use, and that was actually the first utilization of monoclonal antibodies. We're testing, because you can tag then a antibody with a fluorescent protein, right? And then you can do an antibody wash And if your proteins are there and that antibody will recognize it and bind, and then you shine a UV light on it, and then you can, oh, look, the proteins are there. And very, very important in research, in assays, in doing different tests. And so antibodies are, I mean, indispensable in research and in clinical applications to test for positive, we use it in the strep tests, are using antibodies. Anything that needs specificity, we can make antibodies for, and then use them for testing, put them in reagents. But then even we can extend beyond that, and antibodies are used as therapies or pharmaceuticals. We call these biologicals. So a lot of time you'll see, like, you can have chemotherapy, chemo agents, which are going to be chemicals, or you can have biologicals. Biologicals are proteins. And in this case, a lot of them are going to be monoclonal antibody. You can give them usually via IV, so you infuse them. And so we call it IVIG for intravenous immunoglobulin. It can also be gamma globulin. And this can also come from, we can generate it in the lab. We can also get it from donations. So if you donate plasma, you get pooled plasma. So people that, usually that's going to be more for like clotting proteins and that kind of stuff, but you can pull antibodies from that as well. So yeah, the MAB, the monoclonal antibody and antisera, can be used to diagnose different diseases. We use it a lot in the clinical lab for testing, but it's used extensively, not only in research, but in therapies. And so it started out by using, nobody uses mouse antibody anymore because it causes a lot of allergies. Then it went to chimeric, where we used the mouse genetics to make the antigen specificity what we wanted, but then we used human genes to make the heavy and light chain constant regions, which was great for allergies, but still ended up causing, we started making antibodies against the mouse variable regions. So then those didn't work very much. Now we pretty much have humanized or fully human bioengineered antibodies. And so now in the humanized, there are just few mouse genetics to make the variable region, and the human one is fully engineered human genes. And so that those two forms really reduce the chances that allergies are going to come about, or we're going to build up an immunity against them. So anyway, we'll look at some drugs that, and a lot of the treatments that we're going to look at throughout the year will also be monoclonal antibody. But it's kind of fun when you start to look at drugs and how they're named. And if they end in, there's a naming whole nomenclature process for monoclonal antibody therapeutics. So anyway, I just want to bring that up as just an extension of how we can use immunoglobulin or antibodies even outside of the adaptive immune response. So that is what I have for this chapter. And yeah, thanks for hanging in there with me.